Good afternoon. My name is Prajwal Podel, Product Development Scientist at Acro Biosystems. Before I begin, I would like to thank the organizers for holding such a great symposium once again. Today, I'll be talking about Acro's application and structure integrated protein development platform for functional and complex therapeutic targets. To satisfy the rigorous standards in pharmaceutical R&D, Acro has developed the application and structure integrated, or the ASI platform, to produce high performance and high quality recombinant protein products. These efforts are divided here into four key areas: improving difficult to express or DTE proteins, applying structure-based design of products, adopting strategies to stabilize multipass-transmembrane proteins, and lastly, development of antigen-displaying nanoparticles. To begin, I would like to showcase some of the strategies Acro employs to improve DTE proteins in HEC293 cells. Additionally, multiple factors during recombinant protein expression needs to be considered and optimized sequentially for the best quality and yield. At the plasmid design and construction level, factors like promoter strength, copy numbers, integration sites are critical. The codon sequence needs to be optimized as well for downstream consideration like mRNA stability and secondary structures, as well as translational efficiency. After translation, folding issues including aggregation, intercellular inclusion bodies, and degradation issues also needs to be addressed. And for a high yield process, vesicular transport or secretion of a mature folded protein is desired. On top of that, Acro's strategy to improve DTE proteins includes expression of ER resident molecular chaperones, addition of chemical chaperones, implementing mild hypothermia to address folding issues, and to increase protein yield. Acro relies on innovative expression vectors and signal peptides to direct strong protein secretion and expression. One example is expression of the inhibitory Notch ligand BLL3. As you see here, BLL3 contains nearly 20 disulfide bonds. And with Acro signaling peptide and chemical chaperones, the expression was improved substantially from little to no expression to robust levels seen here. Another case where Acro was able to troubleshoot and optimize protein production was in the case of B-cell maturation antigen, or BCMA. Optimization in four areas, including vector optimization, fusion tag optimization, signaling peptide improvement, and finally a stable cell line generation, led to an overall improvement in not only in expression levels, but high batch-to-batch consistency. Another key consideration for Acro's protein optimization ASI platform is the focus on native structural features and folding. An example that illustrates this point is in the design of IL-2 receptor complex construction. In vivo interleukin-2 exerts its pleiotropic activities in promoting the proliferation, differentiation, and survival of mature T and B cells, as well as cytolytic activity of natural killer cells through binding to its heterotrimeric receptor complex. Crystal structure of IL-2 and IL-2 receptor complex reveals that the alpha chain of the IL-2 receptor does not make any contact with either the beta or the gamma C chains as shown. Key components of these IL-2 receptors have varying levels of affinity to IL-2, while the heterotrimer complex has the most potent binding affinity. To design a stable IL-2R heterotrimer in vitro, Acro employed the FC knob into hole design to develop a gamma and beta subunit heterotrimer, while linking the C terminus of the alpha chain to the N terminus of the gamma chain through a flexible linker of specified length to maintain a spatial complex conformation and IL-2 binding property. The complex was purified to greater than 90% purity. And binding study using this IL-2R heterotrimer receptor complex revealed that the IL-2 binding is comparable to the native complex in SPR studies, as well as the IL-2R beta and gamma C construct also showed comparable activity. As controls, IL-2 binding affinity of individual chains of Acro purified IL-2 receptor were determined to also have similar to activity to reported values. Acro's focus on ASI platform to recombinant protein generation extends to multipass-transmembrane protein as well. Acro's typical workflow for membrane protein starts with expressions in insect cells or HEC293 cells, and also multiple expression systems, followed by detergent screening for protein solubilization, leading to multiple rounds of chromatography purification, and finally protein stabilization with detergents or methods like nanodiscs. Multipass transmembrane proteins, or MPs, have low expression level, as with any difficult to express protein, due to incomplete protein maturation, insufficient trafficking to the plasma membrane, and cytotoxicity of the protein itself. On top of that, membrane proteins have complex purification process, that is membrane separation, washing, protein solubilization, and purification. And additionally, inherent conformational flexibility of membrane protein make it difficult to stabilize once separated from cell membrane. Now, historically, a workaround to developing therapeutic antibodies against MPs has been to use peptides in the extracellular loop or extracellular domain. However, MP receptors like GPCRs are increasingly focused on cancer antibody therapeutics, and thus availability of stable and biologically relevant formats addressing complex nature of this multi-domain protein is critical for drug discovery. With techniques like nanodiscs, detergent micelles, engineering stable cell lines, and VLPs, high quality and bioactive full-length MPs are now possible to be produced regularly. One strategy part of Acro's ASI platform uses antigen display nanoparticles in stabilizing transmembrane protein is called eVLPs. eVLPs, or enveloped viral-like particles, are formed by self-assembly of envelope cascade proteins from viruses. Transmembrane protein are constituted in situ with VLPs produced from HEC293 cells. VLPs concentrate conformationally intact membrane proteins directly onto the cell surface, producing a soluble and high concentration of proteins. These are perfect for immunization and antibody screening and display properly folded membrane proteins in their native cellular membrane. Furthermore, eVLPs are 150 nanometers in size, similar to the sizes of most viruses, and are optimal targets for dendritic cell in vivo and surface attachment for phase display. Another MP that Acro has optimized are the claudins. Claudins are a family of proteins acting to maintain a tight junction that controls the interchange of molecules between cells. A subtype, claudin 18.2, is target in stomach cancer. However, it is challenging to purify a biologically active version of the protein with only a few vendors carrying this product. Acro has been able to purify it to greater than 90% purity in the eVLP format, and the bioactivity judged through SPR ELISA showed high performance. Another method in Acro's ASI platform for multipass membrane protein is nanodisc assembly method, as showcased by full-length human CD20. CD20 is present in B cell development throughout, except for early and last steps, and has been targeted by monoclonal antibodies like rituximab and ofatumumab as part of the treatment against B cell lymphomas, leukemias, and B cell mediated autoimmune diseases. Now, CD20 exists as a dimer, as shown in this crystal structure, and multiple reports indicate that two extracellular loops form epitopes that are targeted by antibody or CAR T therapies. However, CD20 is difficult to express and purify. And Acro has successfully purified and stabilized full-length CD20. So high purity of CD20 was first obtained with several rounds of optimization during each steps, including in the expression steps, the extraction step, followed by affinity, and lastly by size exclusion chromatography to yield a pure CD20. Once purified, two methods were utilized to stabilize CD20: the detergent stabilization method and the nanodisc stabilization method. Each method has its own advantages and drawbacks. The detergents can separate the lipid membrane from the protein and result in protein detergent complex. Nanodiscs are detergent-free, non-covalent synthetic models consisting of phospholipid bilayers and membrane scaffold proteins, or MSPs, that stabilize membrane proteins and help retain their biological activity. A big advantage of the nanodisc assembly is that it can be formulated into a powder and also is compatible in CAR-T expression. Membrane proteins in detergents are incubated with the MSPs and detergent solubilized lipids to initiate this nanodisk assembly after detergent removal. Acro's detergent stabilized CD20 performed very well against both the rituximab and ofatumumab, as shown here. And Acro's nanodisk assembled CD20 also performed very well in ELISA assays using rituximab and obinutuzumab. Lastly, Acro Biosystem is a premier recombinant protein supplier, as well as supplier of antibodies, kits, and pre-coupled magnetic beads. Please visit our website at acrobiosystems.com for more information and to learn more about our more than 600 plus recombinant protein catalogs, including COVID-related proteins and variants, immune checkpoint proteins, and CAR-T target proteins. Acro Biosystem is ISO and certified, and some of our kits are also IVD CE certified. My email is proswell.podol@acrobiosystem.com. And for any general inquiry or product information, you can contact us at order@acrobiosystems.com. Thank you for your attention, and I'll be happy to take any questions.